Hi, I'm Heather, and this edition of the Longevizine is for the week of May 18th, 2026. Somewhere in your liver right now, a cell has stopped dividing. It pulled itself out of the cell cycle, maybe months ago, and it has no intention of re-entering. By the definition the senolytic field has used for the last decade, this cell is a problem. It is senescent. It is releasing inflammatory signals into the tissue around it. It is, in the cleanest version of the story, a thing to be cleared. A comprehensive review published in Aging US this month by Deng, Yang, and colleagues at West China Hospital, Sichuan University, tells a less clean and considerably more useful version of that story. The review walks through senescence across the liver, lungs, kidneys, heart, adipose tissue, brain, and skin, and the argument it develops across all of those organs is that senescent cells are heterogeneous in ways the field has been slow to incorporate into its therapeutic strategy. Some of them are doing exactly what the story always said, driving chronic inflammation, disrupting tissue architecture, accelerating the progression of age-related disease. Others are doing essential physiological work, tissue repair, embryonic development, the ongoing maintenance of structural integrity in organs that depend on it. The clinical implication runs through every page of the review. The next generation of interventions has to distinguish between these populations. Clearing senescence as a category is the wrong move if the category contains cells you need. A decade ago, the project was simpler. Senescent cells were zombie cells. The intervention was clearance. Fisetin, quercetin, dasatinib, navitoclax, tools built to remove a population defined as unambiguously harmful. The evidence accumulated and the framing cracked. Some of the cells the framing called zombies were running repair programs the tissue could not afford to lose. The move the field is making now is to keep the original observation, that an accumulation of certain senescent cells contributes to age-related disease, and to rebuild the response around precision identification of which subpopulations warrant therapeutic targeting, and which ones are still earning their place. The instinct was not wrong, only incomplete, and the incompleteness turns out to matter in a way that reshapes the entire project. That same pattern of a binary framing replaced by a more careful reading of which disagreements between the body and its conditions actually warrant which kind of correction runs through most of the rest of this week's research. What follows is an attempt to follow the thread. Two kinds of correction. Laura Koniver's bioelectrical review in current research in complementary and alternative medicine sits at the far end of a very long evidentiary spectrum from the senolytics review, and the two papers together bracket a useful frame. The Koniver review proposes that direct conductive contact between the human body and the earth influences mitochondrial ATP production and reactive oxygen species generation through 5 overlapping pathways: redox status, blood rheology, autonomic regulation, cortisol and circadian rhythm, and inflammatory signaling. It calls this emerging subfield conductive medicine. The review is honest about what it does not have, which is the thing that makes it worth citing. The primary human studies remain unfortunately small. The mechanistic story has more theoretical scaffolding than rigorous, controlled trial data behind it. Koniver treats this as a reason to argue for better studies, rather than a reason to make bigger claims, and the result is a paper that sits comfortably in the emerging register without pretending it has earned the established one. The body evolved in continuous electrical contact with the ground. Rubber soles, insulated floors, elevated beds. The modern built environment removes that contact almost completely. Whether or not the precise mechanism turns out to be the redox-active electron transfer the review proposes, the structural observation underneath it holds. A population that evolved in a particular electromagnetic environment now lives almost entirely outside it. The disagreement is real. What to do about it, and at what confidence, is a separate and interesting open question. A third paper this season speaks to the other side of the same picture. A preprint using the Finnish twin cohort and 2 parallel exercise studies tested whether nicotinamide riboside supplementation and high-intensity interval training produce the same effect on skeletal muscle epigenetic aging. NAD+ levels appear to decline with age in some tissues, and 2 primary strategies have emerged to restore them: a molecule, NR supplementation, and a load, exercise. The preprint finds that the 2 strategies affect different epigenetic markers, They are doing measurably different things at the level of methylation, even when aimed at what looks like the same gap. This is the shape the whole week makes when you stand back from the individual items. The body has disagreements with the environment it currently lives in. Some of those disagreements respond to a molecule applied at the precise point of mismatch. Some respond to a return of an environmental condition the body is still calibrated for. Some of them respond to one and not the other, and the NR versus exercise result is the cleanest demonstration this season of why it matters to know which kind of move a given gap calls for. Applying the wrong correction to a well-located gap is a waste of attention at best. Understanding the shape of the gap well enough to match the move to it is the whole of the skill, and what makes the difference between a scattered pile of health behaviors and a set of specific answers to specific questions. Held lightly. Most of what any of us actually know is what we have experienced directly. Everything else, including the research cited above and the framings developed around it, is a belief held at varying levels of confidence based on the inputs each of us has decided to trust, by criteria each of us has set individually. There is no way out of that condition. Even experience is filtered, sequenced, and named through cognitive structures nobody designed for themselves. What gets called knowledge is, almost all of the time, a working belief calibrated against a set of trusted sources. 3 evidence registers run through every piece of writing in this space: established, emerging, speculative. Applied at the level of each claim, not the article. They are not hedging language. They are an attempt to make the calibration visible so a reader can evaluate the weight and quality of evidence for themselves and reach a different conclusion if the evidence warrants it. Saying that the grounding literature sits in the emerging register rather than the established one is a way of marking how confidently a belief is held in the literature while keeping the belief itself in active circulation. The Senolytics Precision Review reorganized the senolytic story this month. The next major paper may reorganize it again. Sitting with that, without treating the reorganization as either a personal betrayal or a vindication, is most of what makes a long-term practice of self-directed experimentation sustainable rather than exhausting. There is a second point that matters more than the methodological one. A belief held lightly enough to update when new evidence arrives is also a belief that does not have to be defended against people who hold it differently. The most expensive thing a personal health position can do is become a wedge. Conversations about peptides, psychedelics, hormones, diet, light, consciousness, which authorities deserve trust on, which subjects. All of these have polarized considerably in the past several years, and the polarization has real costs that have nothing to do with the underlying science. Friendships strained over supplement stacks. Family dinners spoiled by diet arguments that have more to do with identity than with metabolism. Working relationships walked away from because a disagreement about a protocol felt, somewhere along the way, like a disagreement about character. The position itself was rarely the problem. The weight assigned to it was. A belief that has to be defended at the cost of a relationship is being held at a weight the evidence cannot support, and the excess weight is doing more damage in the relationship than the belief is doing in the body. The work is to hold the position, examine it when a new thought arrives, and keep the connection intact across the disagreement. Most of what gets called certainty in health discourse is, on close inspection, a confidence the evidence may or may not have earned, and information shared without forcing a belief is relationship building instead of crushing, and community is what will save us. Building the world back. The body and the modern environment disagree in a number of specific, locatable ways, and a great deal of modern medicine, outside of acute and emergency care, is structured around catching those disagreements after they have deepened into a diagnosis and applying a correction at the precise point of mismatch. Hormone replacement patches the hormonal clock. The GLP-1 drugs patch the food-environment gap. Vitamin D supplementation patches a light and latitude mismatch. The structure of the move is the same in each case. Locate the gap, apply the molecule. There is a second move available, less often named and harder to build a business around, which is to hand the body back a piece of the environment it was designed to operate in, and let the native regulatory systems do what they were built to do, without needing as many corrections in the first place. This is the constructive version of the observation that human life is somehow mismatched to modern conditions. Several specific tunings of the body's regulatory systems are mismatched to several specific features of the built environment, and most of those features are construction choices that can be made differently. What that looks like in practice is less about protocols and more about the shape of the day. The morning matters more than most of what follows it, because the body's master clock was built to read a signal from the sky that the indoor environment almost never delivers. Bright light, real outdoor-grade light, within the first hour of waking, is the strongest single anchor the circadian system has. The Frontiers in Neuroscience review on melatonin and aging, published this year, makes the case again that what follows from a well-anchored clock, the depth of overnight repair, the timing of hormone release, the synchronization of metabolic processes across tissues, is downstream of essentially every aging process the literature discusses. Melatonin in its systemic role is a circadian synchronizer, a mitochondrial protector, an immunomodulator, and a neuroprotective agent. An age-related melatonin decline contributes to accelerated aging through oxidative stress, mitochondrial dysfunction, and inflammatory burden. A bottle of melatonin from the supplement shelf can have a role here. The window open in the morning has a larger one, as does going to bed when it's dark out. The kitchen is the next piece of architecture that matters, and the leverage in it is friction. The body's appetite regulation was calibrated for a food environment where every calorie required effort to find, prepare, and consume. The modern food environment is defined by constancy, abundance, and the near-total elimination of friction between the impulse to eat and the act of eating. The GLP-1 drugs are doing important and well-targeted work at the receptor level to close this gap. The environmental version of the same move is a kitchen where the friction is deliberately put back in. The time a real meal takes to plan and prepare, the fiber and structure of food that has not been processed past the point where the body's satiety signals can catch up, the physical act of sitting down and eating slowly enough that the fullness signal arrives before the plate is empty. Load does not have to live in a gym. The body requires mechanical stress on bone and muscle regularly across the lifespan, and the most sustainable version of that stress is the kind that accumulates without a willpower budget. Stairs, taken because they are in the way. A walk built into the commute rather than scheduled as a workout. Weight carried through the course of an errand rather than lifted in a set. The gym workout matters. The day that contains incidental load throughout it matters more, because it compounds without the psychological overhead of motivation and compliance. And the research on bone density, muscle maintenance, and metabolic health all converge on total daily mechanical stress, rather than on any particular exercise modality. Recovery is the last piece, and it is the one most often treated as a behavior rather than as a place. The body's stress response was built for an emergency that resolves in 90 seconds, and the modern environment sends a signal that holds the emergency cascade open for hours or days. The stand-down signal the system is built to read is physical, environmental, and social all at once. A room where the noise stops, a body in a horizontal position before the room goes fully dark, a relationship or a solitude that does not require performance or vigilance. Building that into the architecture of a home and a schedule, rather than reaching for it after the fact through a meditation app or a breathing exercise or the expensive bourbon, is the environmental version of the same move, and it is the one the nervous system was built to respond to. None of this replaces the molecule, the supplement, the targeted intervention applied where the architecture cannot reach. It changes the order of operations. It puts the environment first and the correction second, so the correction is chosen and placed, rather than improvised under pressure. The molecules will continue to get better, and the precision will continue to sharpen. The biggest gains, for most people, are still in the shape of the day. Signals from the field. Auburn researchers receive funding for Mitochondrial Dysfunction First Aging Program. A new program at Auburn University, led by Andrew Ross, will investigate mitochondrial impairment as a determinant of aging onset and disease trajectory, phys.org reports. No results yet, and the framing is institutional rather than novel. The signal worth tracking is the steady accumulation of mitochondrial dysfunction first framings inside mainstream funded research. Institutional science is arriving at a framing that practitioner communities have been operating inside for years, and the convergence itself is the story. MIT publishes roadmap on transcranial focused ultrasound for consciousness research. A roadmap paper from MIT's neuroscience program proposes that transcranial-focused ultrasound can stimulate deep brain regions precisely and non-invasively, and outlines a research program that would begin with the visual cortex and move into the frontal cortex, with the explicit aim of distinguishing whether conscious experience requires higher-level cognitive processing or arises from simpler sensory mechanisms. The significance for the consciousness research landscape is methodological. The field now has a tool that can move beyond correlational neuroimaging and into causal intervention, which is the step required to distinguish genuine neural correlates of consciousness from downstream effects. Peptide regulatory architecture continues to clarify ahead of July PCAC meetings. The May BSCG analysis, What's Changing with Peptide Regulation in 2026, and the AgeMD May 12th patient-facing piece are the cleanest current legal walkthroughs of the post-April 22nd regulatory state. The HHS Secretary's April 15th confirmation of 12 peptides removed from Category 2 does not confer Category 1 status automatically, and each substance requires individual PCAC review. For anyone considering a public comment on the 7 peptides under review, BPC 157, KPV, TB 500, MOTSC, DSIP/amideltide, Cmax, epitalon. The operational dates sit in the field notes below. Frontiers in Neuroscience publishes integrative review on melatonin, sleep, and antioxidant strategies in aging. The Frontiers in Neuroscience 2026 volume includes a review synthesizing the case for melatonin as a multifunctional, aging-relevant molecule well beyond its sleep role. The paper anchors the circadian architecture argument developed above and is the standing citation hub for any piece on light architecture, sleep as infrastructure, or the circadian biology of mitochondrial maintenance through the end of the year. Field notes. May 17th, forward. FDA-2025-N-6895, docket open at regulations.gov for peptide PCAC comments. June 3rd to 5th, Quantum Effects in Biological Systems, Cubes, Budapest, Hungary. June 10th to 11th, Quantum Biology, Cognition and Medicine Workshop, Växjö, Sweden, embedded in QIP 26. June 18th to 19th, 8th World Aging and Rejuvenation Conference, ARC 2026, Prague, Czech Republic. June 22nd to 25th, Bio 2026 International Convention, with a Translating Longevity Biology track, San Diego, California, United States. June 26th to 27th, The Longevity Show, London, United Kingdom. June 30th, FDA PCAC Oral Presentation Request Deadline, regulations.gov. July 9th, Peptide comments formally provided to the PCAC for the July meeting, regulations.gov. July 22nd, final deadline for peptide comments to be considered by FDA in subsequent rulemaking, regulations.gov. July 23rd to 24th, FDA Pharmacy Compounding Advisory Committee meetings on BPC 157, KPV, TB 500, MOTSC, DSIP/amideltide, Cmax, and epitalon, Silver Spring, Maryland, United States. Late summer or fall 2026. Expected FDA decision on Compass Pathways, COMP 360, for treatment-resistant depression. Before end of February 2027. Second PCAC meeting reviewing GHK-CU, melanotan 2, cathelicidin, LL-37, dihexaacetate, and one additional substance. March 7th to 12th, 2027. Gordon Research Conference on Quantum Biology, Pomona, California, United States. The Phoenix Lab is where rigorous science meets the questions most researchers will not ask. If you know someone building their own approach to biological optimization who wants more signal and less noise, forward this their way.